So that the movie you were just watching is a wonderful, wonderful animation that's available through the links in the links section for the ICON website. The one I was just showing you was supposed to be The Inner Life of the Cell with music, but I don't know where the music went. It's quite wonderful to watch with music as just sort of a contemplative thing. If you'd like to know more about what's doing what to whom, the narration is also very good. So I can commend that to you also. It is awesome, isn't it? It's just wonderful. The new technologies for visualizing cells and molecules are just beyond anything I conceived of when I was sitting on your side of the podium. I was very happy the first time I looked in a transmission electron microscope and saw actual mitochondria and chloroplasts that I myself had fixed. So things have changed a lot. It was still pretty exciting. Before we get into the, let's see, before I get into more about enzyme kinetics and inhibition, I'd like to, how does this thing navigate? Oh, here we go. I'd like to remind you all that we do have both times to meet with, I have times to meet with you in my office during office hours. Both Tim and Lynn are happy to meet with you as well. If you're not completely happy with how you did on the first hour exam, please don't lose heart. In years past, people have done less than wonderfully on that and still ended up with an A in the course. It's 1 out of 5, so there's lots of time to improve and do better on the next test. If you have any concerns you'd like to air with us, please come and we'll help you strategize. If you're not doing quite as well as you wanted to, I would really warmly recommend to you to take regular advantage of the time that Lynn and Tim make available, or to form a study group, or to do both. Just sort of as a checklist for yourself, if you're not completely happy, you should be asking yourself, do I regularly read the chapter? Do I regularly either come to lecture or catch the video if I had to miss it for some reason? Do I process the material, not just highlighting, but do I think it through and write it down? Do I work through the review questions that are embedded in each chapter? So there's a lot of things you can do to really get a grasp of the biochemistry if you're not completely happy with how you're doing right now. And we're quite happy to meet with you to suggest strategies you might not have thought of yourself. Just in the course of a conversation with somebody else this afternoon, I remember a year or two ago, one student, all it took for him to do well on the Friday exams was to remember to eat lunch. He'd been arriving with essentially no blood sugar, and his brain didn't work. So sometimes it's as simple as that. Sometimes it's things like turn off the TV while you're studying, Lots of things that might be small, might be idiosyncratic. All of us have had experience that we can share with you on what might work for you. So any other questions, either about the TAs or me, or about the last hour exam? And you've posted the essay grades, is that correct? Yeah, the essay grades are posted, so you should be able to see them. And then also posted a sort of an explanation of what she was looking for in the essays. If you still have questions, please feel free to get in touch with Lynn, and she'll be happy to explain it in more detail. Okay, so let's go back to enzyme kinetics and inhibition. Does anybody... Are there any particular questions people want to re-examine before we continue from where we were? Are there any questions from the discussion on Wednesday? Remember, if you have a question, almost certainly one or more other people will have it as well, so don't be shy. You can. If you get X number of points on the bonus and Y number of points on the regular part, and it adds up to over 100, you would have over 100%, but it won't show in your exam thing. It'll be the sum of the two. We have a separate column for the exam and for the bonus credit, because the bonus credit is graded by hand. So there's separate columns for them. But yeah, it would be the same as getting more than 100% for the first test. Other questions? Okay, well then let's roll. So, I think we had gotten to this slide, and just to sort of remind you about this, in the case of competitive inhibition, a competitive inhibitor can only bind at the active site, so it or the substrate, but not both, will occupy the enzyme, and its effects show up as an apparent change in the Km. That is, it looks like it takes more substrate to saturate it, half saturate it, than in the absence of the inhibitor, and that's because some of the time the inhibitor's sitting there. So the only change you see with a competitive inhibitor is a change in the Km, the apparent affinity. In the case of a non-competitive inhibitor, you can have a change in the Vmax, how quickly the enzyme goes when it is saturated, because it's just not as good an enzyme anymore, but it has the same affinity for its substrate. So its Km is untouched. And then we have mixed inhibition, where both the apparent affinity and the maximum velocity are affected. All of these things have relevant examples out there in terms of drugs or herbicides or biological feedback systems. These are all strategies that have been used. And the more complicated ones, like mixed or uncompetitive inhibition, often reflect the fact that an enzyme has more than one site on it where something can bind. So something that acts as a competitive inhibitor at an active site, if there's a second substrate somewhere, it may well act as a mixed inhibitor for the second substrate. But it should also bring home to you that Km, which is more or less a measure of affinity, and Vmax, how quickly an enzyme goes when it is occupied, are two different properties. They are independent properties, and they can be affected independently depending on the inhibitors or the feedback regulators that are there. Any questions about those? Okay. Some of these variations come out especially clearly when you use a Lineweaver-Burke plot. So these are the double reciprocal now. So you have 1 over the substrate compared to 1 over the velocity. And in the case of a competitive inhibitor, as you have more and more inhibitor, the intercept, which is 1 over the Km, is getting in closer to the axis. It's getting bigger. It's a lower affinity. But the 1 over the Vmax has not changed. If you look at a non-competitive inhibitor, as the concentration of inhibitor increases, you can see a change here in the Vmax. It's rising on that axis because that's a smaller number. No, excuse me, it would be, smaller number that way. And that's a sign that it's not acting as good an enzyme when it's occupied. And in the case of the mixed inhibitor, both the Vmax and the Km are now affected. All right. Now, one very strong reason that thinking about these things should be of interest to you is because we see various kinds of what could be called inhibition in the control of metabolic pathways. Especially we see examples of what's called allosteric inhibition. In the case of allosteric inhibition, the activity of this enzyme for this substrate can be controlled by some other compound. This is just one other example, one very prominent example of it. We're going to be talking about this particular example in more detail when we get to metabolism. Here you have a substrate called fructose 6-phosphate. So it's a 6-carbon sugar with phosphate there and phosphate there. And we're going to add another phosphate through an enzyme called phosphofructokinase. Now, on the principle that enzyme names often tell you what they're doing, this is a kinase. It's going to add a phosphate, and it's going to do it to phosphofructose. So phosphofructokinase is going to add a phosphate to this phosphorylated sugar. And here's the product, fructose 1,6-bisphosphate. So there's now a phosphate on the 1 carbon, still has its phosphate on the 6 carbon. And it releases ADP at the end of it. So this is the enzyme phosphofructokinase, or PFK. There are a number of other steps before you get to phosphoenolpyruvate, which is the last compound in glycolysis. And glycolysis has binding sites on phosphofructokinase that change the activity of the enzyme. In fact, they negatively regulate it. So as the concentration of phosphoenolpyruvate rises, there's feedback inhibition that goes back, slows down the rate at which phosphofructokinase is producing fructose 1,6-bisphosphate. And you can see how that would be a very good general strategy, that when the product at the end of a pathway can control further, near the beginning, you can keep from wasting energy by putting more stuff into a pathway where there's enough around for your current purposes. And this is also an interesting part of it. The way in which phosphofructokinase is affected is quite striking. PEP is for phosphoenolpyruvate. PFK is phosphofructokinase, the enzyme. And here is an enzyme activity graph for phosphofructokinase. Here's V0 over Vmax. And here is the concentration of substrate here. And in the absence of PEP, the enzyme has a curve like this, which is our classical hyperbolic kinetics, or hyperbolic curves that we see with things like myoglobin. And in the presence of phosphoenolpyruvate, there's a major shift in the Km. The apparent affinity is quite different. Or the affinity, I shouldn't say apparent affinity, the affinity. So that it takes quite a lot more fructose 6-phosphate to get that level of activity. However, if you look here, the Vmax is not really affected. So it's changing apparent affinity. It's changing the shape of the curve. It now has sigmoidal kinetics instead of hyperbolic ones. And it turns out that we can connect back to what we were thinking about with myoglobin and hemoglobin. Phosphoenolpyruvate makes PFK into a tense state in which it's harder for it to bind the substrate, fructose 6-phosphate. ADP helps to keep it in the relaxed state. Now, that might be a little bit counterintuitive. If you look back here, ADP is one of the products of this enzyme. It's one of the products of the enzyme, and yet I'm telling you that it keeps it more in the relaxed state. It can counteract to some extent the PEP. That's because ADP is also a marker for the fact that the cell is short on energy. So PEP is a much better feedback inhibitor because it's more specific to the current status of glycolysis. ADP is a very generic or broad spectrum signal. It's a signal for the energy state of the whole cell, and so it actually has a meaningful effect on the enzyme. It keeps it in the relaxed state. Well, what are some other ways that we can alter enzyme activity? It turns out that cells are amazingly ingenious, and almost anything you can think of to do to an enzyme that might change its activity has probably been done by a cell. You can change synthesis or degradation rates. Now, in general, in living systems, synthesis and degradation are sort of the two sides of the coin. If you increase synthesis, it's the same thing as decreasing degradation. You can get more of a product either way. Or if you decrease synthesis, it's the same as increasing degradation. You have less of that product. There's a real yin-yang about this. So if you change either one of those, you're going to change enzyme activity. And a very important example of that is for one of the enzymes that's involved in making new sugar out of bits and pieces of fatty acids and amino acids. One of the major ways in which that enzyme's activity is regulated is a signal that turns on transcription in the nucleus for more translation, so it actually makes more protein. We also see that some of the hormonal changes are done through this kind of a thing, because things like thyroxine have rather a long-term effect on gene regulation and enzyme activity, and they do that through changes of that kind. You can change location. And so this is sort of a little schematic for a change of location, where you can see something that's in a vesicle to begin with can be moved out to a new location where it'll be effective. And we're going to see a concrete example of that in the next chapter on membranes. You can control enzyme activity through ions. It can be a fairly rare ion like calcium. It can be something like as simple as a proton, pH. And they're giving you a little diagram in here showing you how the endoplasmic reticulum is a store for calcium, and that by its triggered release, it can turn on or turn off enzymes in its neighborhood. There are many examples of covalent modifications. In this case, they've shown you one where they phosphorylated it. Phosphorylation can turn an enzyme on. It can turn it off. There's cases where we see modification with lipids. You actually add covalently a lipid. Now, you might think the phosphate group is a fairly small thing compared to the size of a whole protein. Isn't it odd that one little phosphate group can turn on or turn off an enzyme? But if you think about a doorway and you think about how tiny a doorstop is, one little wedge of wood can keep the entire door open. And so the correct placement of the right thing in the right spot can have a big effect on a larger thing. Any questions? Okay. So these are thought questions, and I don't know whether I should confuse you right away by saying there aren't necessarily right answers or not. But I think it's interesting to realize you can ask questions that don't necessarily have clear answers. So if you take the simplest imaginable case, a real diagram type, a real cartoon type enzyme, if you were to change the catalytic residues by mutations, what would you imagine might be the most likely target for its kinetic variables to be changed? Okay, did you say KM? Do you want to discuss that a little? Okay, so you're saying KM is a measure of affinity, and if you change the residues, you may well change affinity. In the real world's kind of enzymes, that's actually probably a correct answer. That is, the catalytic residues may well affect affinity as well as catalysis. And so I think that's important to keep in mind, is that the catalytic residues are not just doing catalysis. They are doing catalysis, but they're also involved with binding. There are examples, I can't give a number for how many examples, where you can change one of the catalytic residues and knock out catalysis completely, and yet retain substrate binding. And I think you can see how that might happen. It won't necessarily happen every time, but you can see how it might happen, that you just knock out one key proton for the reaction overall, but so much of the rest of the surface is still the right shape to bind things that it can continue to bind them. It just can't do anything with it now that it's there. I think you can see how this kind of reasoning is just sort of flipped for the specificity sites, things like the side chain pocket in chymotrypsin. And again, it's one of these things where in the real world you could have either thing happening. You can see how changing that specificity pocket could change specificity only, because you can see that in chymotrypsin and trypsin you have similar catalytic abilities. They're just choosing different things to do it to. But it can also be the case that if you change the way in which something binds, you then change how catalysis proceeds. And so you could have a change in Vmax as well from the specificity site. So let's look at some inhibitors that are of great interest these days and are playing a key role in parts of the world where AIDS is especially prevalent these days. And So HIV stands for human immunodeficiency virus. It's a virus that can, in effect, knock out a significant part of the immune system, and the person who has it then has AIDS or acquired immunodeficiency syndrome. And really what they suffer from when they actually are suffering is more the diseases they get because they haven't got a competent immune system than AIDS per se. And the source of all this trouble and trauma is a virus. It looks kind of like this on an electron micrograph. This is a false color to make it a little more visually striking. It's a single-stranded RNA virus. So there's one strand of RNA that codes for everything that's going to happen downstream from that. Just one molecule of RNA in the same strand. And this core area here has that RNA molecule. It has enzymes. It has proteins that stabilize things and coat them and stuff in there. And then around that core, there is this coat. And in the case of HIV, that coat includes both proteins and lipids. Viruses differ a lot in this. Some have some of the host membrane, some have pure protein. In the case of HIV, it's both proteins and lipids around that central core. HIV has a very cute trick that it pulls. I mentioned that it has a genome of RNA, a single strand of RNA. And if that's all that there were, it would probably not do anything terrifically challenging to the host's genetic sequences. But it has, in fact, in it an enzyme called reverse transcriptase. And reverse transcriptase has a couple of activities which are sneaky. So it can take a copy of RNA and synthesize DNA from that. So now you have an RNA-DNA hybrid here. Here's the original RNA molecule here. Here's the newly synthesized DNA. It also then has an RNase activity that will degrade the RNA that's in the hybrid and leave behind a DNA strand. And the DNA synthesizing activity is sufficiently promiscuous that it'll work off of the DNA transcript as well. Now you have a double-stranded DNA source of information. So this saves it a fair bit of weight. It's packing just the one strand of RNA, but it can reconstitute a complete double-stranded DNA helix through the activity of reverse transcriptase. Turns out that reverse transcriptase may have borrowed some of its enzymatic activity from the enzyme that we all have in our cells that's involved in keeping the telomere ends long enough. That there's a very little of a rather specific reverse transcriptase activity whose sole job is to keep our chromosomes from crumbling. So we ourselves don't have the enzyme reverse transcriptase. That's a viral enzyme. And so that would seem to be a good target for an inhibitor. If you could inhibit something that only the virus has and we don't have, it's usually going to be more specific than something that we share in common. And so in the early days, a lot of the focus for inhibitors for HIV focused on reverse transcriptase. And they took a couple of forms. So there were some that produced or were substrate analogs. So here is an analog that sort of looks like it could be the thymine precursor for the DNA, but you can see that instead it has this azeto group here, and when it gets incorporated, it stops further elongation. Similarly for this dideoxycytidine, it doesn't have any OH there, and so it can't be elongated off of. It's like the dideoxy terminators that are used in sequencing. And so both of these would then tend to frustrate the reverse transcriptase so that it only makes fragmentary transcripts. There are others that are not analogs of nucleotides, and yet that seem to inhibit reverse transcriptase. And so here's an example of that. And you can see from looking at the shape of that that maybe it has a homology to a base pair rather than to a single base. And so maybe what it's doing is intercalating and distorting things that way, or maybe slipping into the site and looking like it's a base pair when it shouldn't be there. Now, HIV has a trick that many viruses take advantage of. Since they're trying to make do with as little nucleic acid as they can, they're packed full of strategies for extracting as much information from that genome at once as they can. And one of their strategies is to use polyproteins. Once you have the double-stranded DNA, transcripts are made out of that for protein synthesis. That RNA transcript is translated by ribosomes into what are called polyproteins. So, for example, this lavender one is a polyprotein that actually encodes 3 enzymes. So the protein will have to be cleaved there, and that will release 3 distinct enzymes that the virus can use either in replication or processing or something like that. Here's an example of a structural polyprotein. When it's cleaved, it'll make 4 different structural proteins. The blue ones are various kinds of accessory proteins. This one's a polyprotein and needs to get cleaved to be active. Here's another structural protein that has to be cleaved to be active. So this gives another target, because for the most part, we as mammals and eukaryotes don't do polyproteins. That's not a very common thing to see in the eukaryotic genome. And so one of the enzymes that HIV codes for is for its own protease. There's a special protease whose sole job is to cleave those polyproteins at the right site. And that, again, is a target that's not exactly present in us. We know enough from looking at the enzyme mechanisms of proteases that it's probably not utterly different, but that specific protease is an HIV gene. And the HIV protease inhibitors were one of the early examples of what's called rational drug design, where instead of simply doing what I was taught in my botany classes, spray, pray, and weigh, where you spray a bunch of things, you pray that something's going to have an effect on something, and then you weigh it at the end to see if it actually did. Pharmaceutical companies were doing spray, pray, and weigh for quite a long time, and oftentimes they got something. But in the case of rational drug design, you actually try to think ahead of time what ought to be a reasonable guess at a transition state analog or something that would interfere otherwise. And so these protease inhibitors try to mimic a tetrapeptide that is in many of the HIV targets. There's a phenylalanine that's important to recognition in a number of these groups, and there's a proline that's next to that phenylalanine. And so you can see in these protease inhibitors, so this is the phenyl ring here next to the proline. That's that fused chain into the peptide nitrogen. Here's a phenyl ring, and it's got a nitrogen near it. It's not exactly the fused ring. Here's a phenyl there, and again, a little bit further down, the fused nitrogen ring. But what they have found is that bulky group there, where the phenyl group would be in the target, is important. Once you have something that can do something in the way of inhibition, you can do a number of things to tweak the edges to make it more soluble or more easily taken up by the cells. Interestingly, even though these were designed for the HIV protease, most of them still have side effects on our bodies, and maybe that's reflecting the fact that there's only so many different ways to cleave proteins, or maybe the side effects are targeting something completely different. So let's work through a few examples of how enzyme kinetics can work. Take a look at figure 36. So, which of these 2 enzymes has the highest affinity for the substrate? You can take a moment to think if you like, or to gather your courage. Which one has the highest affinity? Anybody want to venture? Thank you. Thank you. So A has the highest affinity because it takes a lower concentration to saturate it. Affinity and Km can be a little bit confusing because we speak of being high affinity, having, you know, an avid grip on things. And the way that shows up is it doesn't take a very high concentration to saturate you. So if we take this as an approximation for its Vmax over to here, half of its Vmax would be about here. This would be somewhere about its Km. If we were to take a look at enzyme B and say that this is somewhere around its Vmax and go back over here, go around here to half of its, somewhere around here will be its Km. So clearly, enzyme A needs less substrate to half saturate it. It has a greater affinity or a higher affinity. Which of the two has the largest K cap? B. Would you like to explain why you think it has the largest K cap? Right. So K cat is directly proportional to Vmax. And since this one, under these conditions, has the highest Vmax, it presumably has the largest K cat as well. It can turn over the most substrate molecules per second. Which of the two enzymes is probably allosteric? Did I hear a B? Does someone want to explain why they think B is the allosteric enzyme? It's sigmoidal. So in other words, it sort of has a shallow pitch. It rises more quickly and then tapers off again. And we've seen, both in the case of hemoglobin and in the case of phosphofructokinase, for example, that a sigmoidal curve often means there's some kind of allosteric behavior going on. Allosteric, again, being binding somewhere else, changes the activity of the active site. Which of the two is going to be most active when you have 2 micromolar substrate? A, right. Which one will be most active at 8 micromolar substrate? B. So what we see here is a very concrete example that Km and Vmax are independent variables and that the best enzyme for a particular situation isn't just determined by its kinetic properties, but also by the situation it finds itself in. If the cell doesn't have very much of S, enzyme A will be the best at making more of its product. If the cell has a lot of S, enzyme B would make more product. So even knowing the Km and the Vmax, you don't know which one is the most useful enzyme for this particular cell unless you also know the usual concentration of the substrate. Okay? Any other questions about that one before we move on? Okay. So what do you think the axes here probably are? Looking at a graph like this, given that these are all enzymes, what do you think the axes are probably going to be labeled? If someone wants to raise a hand, I can point at you, make you feel that much more in the spotlight. Yes, please. Thank you very much. So the x-axis here is going to be 1 over the substrate concentration. This axis will be 1 over V naught, or the velocity. Okay. So which of these enzymes has the greatest affinity for their substrates? If you need a moment to think this through, take a second. Anyone want to take a stab at it yet? Which one probably has the greatest affinity? Yeah. Sorry? You want to explain why you think E2 would have the greatest affinity? So enzyme 2 is the purple line. It's this intersection is going to be 1 over its Km, right? So this is 1 over its Km. But unfortunately, this is a reciprocal graph, right? So this is the largest number. Okay. You're right that this is the place you're going to look to try and find the Km, but you have this little tricky thing that because it's a reciprocal graph, this is a larger number than the others. So this one has the smallest Km, and the smallest Km means the greatest affinity. Okay. Again, we have this thing. I don't know whether biochemists should have tried to break themselves of a habit, but we think of high affinities, and yet we talk in terms of Kms when we want a number. So, which of these has the greatest Vmax? Sorry? Enzyme 3? Okay, what would you like to explain your reasoning? Mm-hmm. Right. So this is the axis that's 1 over V naught. Closer to the origin is the larger number, so this is going to have the largest Vmax. Would any of these be very likely to be allosteric? Any thoughts? Since in each case it's made a straight transformation, none of these are probably allosteric. They probably have hyperbolic kinetics, and it's unlikely, not a dead certainty, but unlikely for them to be that. Here's an example of a structural analog. So acetylcholine is a neurotransmitter. And here is decamethonium, which is an inhibitor of the nerves that it works on. Would you expect this to be a competitive or some other kind of inhibitor? Look at how much structural homology there is here. It's, in fact, a competitive inhibitor for its target. Which of the kinetic variables have been changed for this enzyme? Which of the kinetic variables, Km, Vmax, or both, have been changed for this enzyme? You can talk about it to each other if you want. Sorry? Sir, someone have a question about it? Anybody want to take a stab at which kinetic variables are changed for these enzymes? Both of them changed? Okay. Would you like to explain your reasoning for why you say both of them have changed? Yeah, I think given the limitations of graphical methods, the Vmaxes are probably different. This is probably its Vmax, and we can assume it. So if you take this one over here, this is about its half Vmax. That would be its Km, compared to here, which is going to be half maximal about there. Sorry, it's half maximal. That's its maximal. Here's its half maximal. So there's its half maximal down to there. So in this case, both the Vmax and the Km appear to have been changed. Here's an example of inhibitors being good structural analogs for the transition state. So here is a proline and a peptide, and it turns out to be necessary to sometimes to switch the configuration of that proline. And there's an enzyme called proline racemase that can do that, can rotate it more quickly than will happen just by thermal agitation. And here's an inhibitor of that enzyme, and you can see how it would look like a transition state intermediate for that residue. I'm not quite sure why I had that one in there. I don't really know why I included that one. Let's skip over to the next one. There's a question in the book if you want to take a look at it. Okay. This is actually another quite significant enzyme. Aspartate transcarbamylase is an enzyme we're going to encounter later on in metabolism that's a fairly central one. And this is showing you some of the complexity of its regulation. Here in the center line is, if you like, sort of a ground state for its activity. Here is in the presence of ATP, and here's in the presence of CTP. And so you can see that the three of them appear to have about the same Vmax, given the limits of graphics. They've all stayed sigmoidal, unlike the effect that PEP had on phosphofructokinase, where it could change it back and forth. But the affinities, the halfway points, have been moved to the left or to the right. So in this case, the effect of ATP is to move the curve as a whole to the left, or to give it a higher affinity, a smaller Km. Whereas in the case of CTP, the curve as a whole moves to the right, and now its halfway point is about here. It's a larger Km, a weaker affinity, or a smaller affinity. So these are allosteric regulators that can modulate the affinity of the enzyme both up and down. So you could call this one an allosteric effector and an allosteric inhibitor. And this is a case that we're going to be coming back to when we get into metabolism in a big way. Phosphofructokinase, as I said when we first talked about feedback inhibition, is a very, 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 very important enzyme. Maybe you know how I feel about it now. And it turns out to have multiple allosteric regulators. And this is just giving you a little taste of some of the different things. And what you could be asking yourself, for example, is, you know, are these things changing the Vmax or the Km? Are they changing the shape of the curve? And so on. This is fructose 2,6-bisphosphate. It's not a substrate. It's just an allosteric regulator. Look at what tiny concentrations can change the behavior of the enzyme. So is it, in this case, we're looking at two things at once. This is a substrate for it, fructose 6-phosphate, and we're looking at its velocity as a change in its own substrate concentration. Here, we're seeing sort of two kinds of effects at once. If we just look at the first curve by itself, we're looking at enzyme activity again on the vertical axis now. And I already mentioned that ATP is a substrate for phosphofructokinase. Kinases usually are going to need ATP. And so the enzyme activity starts to rise with substrate, and then it plummets. So here's a compound that is a very strong feedback inhibitor. I should call it feedback. Scratch the feedback on that. It's a very strong allosteric effector because it's a substrate for it. It's not a product of it. So the substrate is turning off the enzyme. The more you get more than a certain amount of ATP, and it turns the enzyme off. It's a little odd. And then we see that that effect of ATP is itself changed through this other allosteric effector, fructose 2,6-bisphosphate. The fructose 2,6-bisphosphate can almost completely relieve the inhibition due to the ATP. So this is a way of turning the enzyme back on in the presence of the ATP. Have a good weekend.